his work was really was pioneering in terms of developing these three-dimensional structures, developing and engineering ways to engineer these spike proteins. But let me let him explain his work in his own words. But this is really groundbreaking stuff, and it's because of that kind of work. Yeah, people like Jason, that we were able to develop this vaccine in such a short period of time because they have been doing their work for a very long time. Jason, can I impose on you to say a few words about your work and the foundational aspect of it?
Absolutely, and thank you very much for that warm introduction. You actually had a great explanation for the role that we played in the development of the vaccines. So my lab is a structural biology lab, and where you try and determine these three-dimensional models of viral proteins. And so this is the SARS-CoV-2 spike protein. It's a trimeric molecule made up of 3 copies of the same protein, green, blue, and red. And this is what sits on the surface of the virus, and it attaches to host cell receptors on our cells. And then this protein undergoes a large conformational change to change its shape in order to fuse our membranes with the viral membrane and allow the genome of the virus to enter the cells, reprogram the cells, and start making more copies. And so what we've done is, by determining three-dimensional structures of these spike proteins, we can then go in and rationally engineer in changes, substitutions that we can make to the protein to lock it in this conformation, to prevent it from changing shape. And that way we train our immune system to recognize the specific shape of the molecule, which is the same shape on the surface of the virus. And we raise antibodies against this form of the spike protein. And then when we see the infectious virus, when somebody coughs or sneezes, our immune system already has these antibodies made that directly recognize the active form of the spike protein. And so our work just involved changing 2 residues in this entire spike protein. And that's now used in the Pfizer-BioNTech vaccine, the one from Moderna, Novavax, and Johnson & Johnson. So we've been really excited to see some of the fundamental work, some of the engineering we did in our lab end up being used in 4 of the leading COVID-19 vaccines.
Jason, thank you for the groundbreaking work that you're doing in terms of the COVID.
No, thanks very much for having me, and I'm happy to do this again sometime.